So good afternoon, everyone. Thank you very much for the nice introduction and the BRCCH for organizing this nice event. So as mentioned, today I will present you my work, which is based on the development of a cost-effective device for minimal invasive blood sampling. But why blood sampling? So blood sampling is the basis for around 70% of medical decisions. It is useful to diagnose and monitor disease, but also to check organ health and the hormone levels in the body. There are three main procedures by which we can collect blood: venipuncture, finger pricks, and the recently developed microsampling devices. A bit more in detail, with conventional venipuncture we can collect high blood volume from the vein, which can be used to perform multiple analyses. However, this technique has many drawbacks. It's painful, it's invasive, painful and distressing, not only for children, but in general for all the people struggling with needle phobia and needle fear. It is relatively costly, healthcare specialists are required, and then it results in the generation of a large amount of biohazardous wastes. And then, due to the handling of hypodermic needles, many needle stick injuries are reported every year, which further increase the costs of the procedure. And finally, if we also consider the low-middle-income countries, this procedure is quite challenging due to the lack of healthcare facilities. So, due to all these challenges, finger sticks, and also due to the recent pandemic, finger sticks combined with point-of-care system have become increasingly popular. Of course, compared to venipuncture, they are less invasive, they are easy to use, and they allow to perform real-time analysis, and are also more cost-effective. On the other side, they still have few drawbacks. First of all, the high drop-to-drop variability of the finger sticks, and for example, as we can see here, in dried blood spot, the blood spot is affected by the hematocrit level, because higher is the hematocrit level, which means higher is the viscosity of the blood, and smaller is the blood spot. And this bias in the spot can affect the analyte quantification. And on top of that, the limited amount of blood that is collected with the finger sticks is not always compatible with the point-of-care system that are already on the market. Nevertheless, finger stick procedures are still the preferred choice over venipuncture, as we can understand from this survey study, where 28 children out of 32 choose finger pricks over venipuncture, possibly at home, and due to the lower invasiveness of the technique, they were also very comfortable in self-pricking their own finger. So that's why the microsampling market is, and also due to the need of healthcare decentralization, the microsampling market is projected to grow by the 2030 at an annual rate of around 7.4%. As a proof of this tendency, few microsampling devices have been reached already the market, and they are based on a different technology compared to the finger stick procedures. What makes them so promising is the ability to collect a higher blood volume, which can be used to perform more than one analysis with a collected sample. And of course, they have also the further advantage of mitigate the collection error of the finger sticks. They have as well a few drawbacks. The main limitation is that they are too expensive, especially for the low and middle income countries, and most of them don't allow a direct integration with the point of care system that are on the market. So, based on this overview, our aim was to develop a versatile and more cost-effective blood microsampling device that, after blood collection, can be delivered to laboratories for analysis or can be combined on-site with the commercially available point-of-care system for real-time analysis. The design of the device was inspired by the leeches. So leeches are blood-sucking parasites which are able to attach themselves to the host, bite through the skin, and collect a quite high blood volume in a short time. But how does the leech attach to the host? So he does by using this suction disc, which is located in its anterior part. Now, translated to our device, this feature is here and is called suction cup. Now, back to the leech. In this anterior disc, we also have 3 cutting cartilaginous plates, which are responsible for biting the host. And in our device, they are represented by a concealed microneedle patch array, which is located in the suction cup. Again, to allow continuous blood sampling, the leech uses hypobaric pressure, which is generated through its muscular structure. And as well, we also provide our device with the ability to generate a negative pressure due to the elastic self-recovery of the material after compression. And finally, to avoid blood clotting, the salivary gland of the leech secretes uridine, which is an anticoagulant molecule. And we also provided our device inside the storage compartment with an anticoagulant molecule. So all these features at the end result in the initial structure of our leech-inspired prototype. For the fabrication, we select a fast and cost-effective procedure, the cast molding, by using commercially available silicone. So very briefly, the design was split into the middle to generate 2 negative stamps and molds. And then fresh silicone was poured into the mold, and then the stamp was inserted for the inner structure. After silicone hardening, the stamp was removed, and then 2 identical molds were merged together by using again fresh silicone, and this led to the final silicone-based device. Now, the performance of the device was tested by measuring the adhesion strength with a texture analyzer. So, and at the beginning, we already saw 2 main drawbacks of our device. The first drawbacks referred to the application procedure of the device, and specifically 2 compressions were needed. So, a first compression of the suction cup to fully expose then the microneedle patch, and then a second compression of the storage compartment to generate the negative pressure, which should assist blood sampling. And then the second limitation instead concerned the performance of the device and specifically the low adhesion strength. Now, the adhesion strength gave us an indication how strong the device attached to the skin. In this case, we use a porcine skin. And it also correlates with the negative pressure that can be generated by the device at compression. So our target value was around 50 kilopascal, which was already reported to facilitate blood sampling when applied to the area close to the puncture site. And our value was, as you can see here, lower, much lower. So then, how did we address this issue? So application-wise, we just move the storage compartment on the top of the suction cup. And this resulted in device number 1. And as you can see here from this cartoon, now a single compression of the device was able to allow the exposure of the microneedle patch and also the generation of the negative pressure after the elastic self-recovery of the material. Instead, in terms of performance of the device, we optimized the design, working on different parameters. First of all, the geometry of the storage compartment, so this upper chamber. And by using simulation, we compare the strain energy distribution profile of 3 different devices. So the strain energy distribution can be correlated to the energy that is stored in the material at compression, and specifically higher is the value, which means more yellow reddish in the color map, then higher will be the performance and then the negative pressure generated. Device 3, as you can see, was the one showing the best results in terms of distribution of the strain energy, but also it resulted in a lower residual air in the lateral pocket compared to device 2 and 1. And experimentally, we determined once again the addition strength of all these devices, and then we found that device 3 together with devices 4 and 5 were showing the highest value of the addition strength. And for this reason we selected this device for the second optimization step. In this case we looked at the effect of the silicon hardness. And again, through simulation, we learn that by increasing the shear hardness of the silicon from 28 to 50, there was an increase in the strain energy distribution profile, and as well as in the performance of the device. And the same trend was also observed through experiments for almost all the devices, and specifically then we selected device number 3, fabricated by using silicon with a shear ray of 50, because it was resulting in the highest value of the addition strength. At this point, we moved to the third optimization step, where we investigated the effect of the bottom angle of the suction cup on the addition strength of the device. And as you can see, a bottom angle between 103 degrees and 120 was really necessary to have a good suction of the skin inside the device, and then to really have a good adhesion of the device to the skin. Again, between these two devices, we continued working with device number 3, which was selected for the last optimization step. And in this case, we looked at the effect of the wall thickness. And again, through both simulation and the experiments, we optimized the wall thickness of this device, and we obtained for device 14 our target value of addition strength, around 50 kilopascal. Now, we were able to generate our target value of the addition strength, but what about the real negative pressure generated by the device? We measure it by using a customized setup, as you can see here. So the setup consists of a 3D printed housing on the top of which a silicone substrate was glue to mimic the mechanical properties of the human skin. And then we drilled a little hole on the top of the silicone substrate connected to a pressure sensor. After manually compressed the device on this platform, we measure the negative pressure, which results to be around 74 kilopascal. Now, it's important to consider that this pressure is a combination of two main contributions. The first one is the real negative pressure that is available for blood sampling, and the second contribution is the pressure that is responsible for the skin suction inside the cup. And we wanted to discriminate between these two contributions. So to do this, we work in collaboration with the mechanical department at ETH Zurich, and we use a nimble setup equipped with different pins having a fixed diameter equal to the diameter of our device and different heights. And we measure the pressure required to obtain a specific displacement of the skin inside these little pins. And we fit the experimental data into this exponential curve. After that, we tested our device ex vivo on porcine skin, and after sectioning, we determined that our device was able to suction the skin around 5.8 millimeters. Now, substituting this value into this equation gave a pressure responsible for the skin suction of around 14 kilopascal, which means that the residual pressure of around 60 kilopascal is the real pressure available for blood sampling. And compared to other microsampling devices that are already on the market, this value is a little bit higher, which could suggest a potential better performance. After the optimization of the cup, then we moved to the fabrication of the microneedle patch array, and we tested 2 different stainless steel microneedle blades, and we used both of them to prepare rectangular or circular patches, which were tested ex vivo. We found that circular patches resulted in a more homogeneous and also deeper penetration depth into the tissue, and then, however, only the microneedle blade number 2 was able to require a lower penetration force. That's why we select at the end the circular patch composed of microneedle blade number 2 for the final device assembly. To assemble the device, we slightly modified the design of device 14, just inserting a cylindrical dome here to allow the insertion of the microneedle patch. And we also provided the device with a 3D printed adapter in order to facilitate the compression of the device and then its application. And we also provided the device with a skin adhesive here to form an airtight seal with the skin. 2 different prototypes were realized with 20 or 30 microneedles. And after that, we tested the performance of our prototype in vitro. So we used this customized setup, a kind of France cells, inside of which we insert different sampling media. And then on the top of this France cell, we glue a silicone platform, again to mimic the mechanical properties of the human skin. After manually compress our device on the setup, we estimated that in the most complex scenario, our device was able to extract around 500 microliter of porcine whole blood. And then, since our device was supposed to be delivered to laboratories for analysis or combined on-site with commercially available point-of-care system, we also designed and 3D printed customized adapter or lead for the different application. And in the case of combination with point-of-care system, we also test this kind of feasibility. And what we did was just to load into the storage compartment of the device 500 microliter of human blood spiked with recombinant Plasmodium falciparum histidine-rich protein 2, which is a marker for malaria detection. And after assembling the device with the adapter, we were able to extract a single blood drop on the commercially available malaria pepper strips, hence confirming the feasibility of this type of combination. And finally, we also tested the performance of our device in vivo, in piglets, by applying the device on the lateral flank of the piglets for 10 minutes. And then by weighing the device before and after application, we determine the amount of blood that was collected. What we saw is that the device with 30 microneedle performed better compared to device with 20 microneedle, and we also found that when the device was applied in the central lane of the flank, the performance was improved, suggesting that also the specific body location can suggest a possible better performance of the device itself. And finally, to also confirm the minimal invasiveness of the prototypes, we took a photograph of the piglet skins at different time points after the removal of the device, and after 24 hours, as you can see here, there are only few application traces that are visible, confirming therefore the low invasiveness of our prototypes. And this brings me to the conclusion. So we develop a minimal invasive device for the collection of capillary blood to enhance the patient compliance. So the device design and the microneedle patch array were optimized to build a device with an increased performance, which in vivo was able to sample around 200 microliter of blood, which is an amount already compatible with many point-of-care systems. And since the design is very simple, this can also, the fabrication of the prototype may enable scalability and also suggest a potential cost-effective production. And at this point, we are working on the development of a fully biodegradable version of the device to further address another issue that is frequent in the low and middle income countries, which is the waste management. And with this, I would like to thank my PI, Professor Jean-Christophe Leroux, all the people that work with me on this project, our collaborator from the Mechanical Engineering Department, and of course the Borton Research Center for funding this project, and you for the attention. Thank you.